Welcome, everybody. My name is Chloe, and on behalf of Oxford Global, I would like to thank you all for joining us for today's webinar, A New Golden Age for Target Identification, Accessing the Novel Druggable Space with Discovery Proteomics. I'd like to introduce our two speakers for today, the first being Dr. Mo Jain, founder and CEO of Sapient.
Thank you so much, Chloe. Really excited to be here today and to discuss as to how proteomics is transforming target identification and validation.
And we are also joined by Dr. Jonathan Yusuka, Chief Business Officer at Sapient.
Thanks very much, Chloe. Very much looking forward to the discussion. We see a lot of changes in the landscape of target identification and a lot of opportunities for proteomics to contribute.
Thank you. We're very excited to host this presentation today and invite you to submit any questions you have throughout the session via the Zoom chat to be addressed during the end Q&A. With that, Mo and Jonathan, please take it away.
So to focus today, we're going to be talking about how drugs treat disease and what are the major trends in how that is changing. I'm Jonathan Yusuka. I've spent the last 25 years as a drug hunter. My entire career, we've been looking at a relatively small number of proteins. So the total number of human proteins is about 100,000 if you include all the forms of them, or about 20,000 if you stay canonically linked to the genome. Of those 20,000, only about 4,000 under current technologies are considered druggable. And to date, we've only really had medical traction with 900 of those proteins. So on the graph on the right, you see the x-axis shows different individual targets, and then the number of NMEs that have been approved against those targets to treat disease. And you see that we have remained highly concentrated on a small number of targets. So most medicines are coming from the pursuit of a relatively very small number of targets. What we want to talk about today is why we believe this can change based on our technologies, the technologies that are coming to bear in the pursuit of drug development. So first, why have proteins been difficult to drug traditionally? I just mentioned that there are about 20,000 to 100,000 different forms of proteins, and only 900 or so have actually resulted in drugs. The druggable targets traditionally have been things like enzymes or transporters or gene-protein coupled receptors or CD markers, etc. These have very well-defined active sites and binding pockets. The remainder of proteins, while very much involved in disease and even causing disease, have certain structural properties that have made them traditionally difficult to drug. So they have things like they don't have a well-defined binding pocket, or the binding pocket is too shallow, or that binding pocket is non-specific. It is shared by a number of different proteins. And so you start to get a proliferation of off-target effects. Or the function of the disease aspect is coming from a very complex set of protein-protein interactions. So for all of these reasons, we've been highly limited by the number of drug targets that we can pursue pharmaceutically, which results in that cluster that you saw on the previous page of virtually all pharmaceuticals coming from a small number of targets. But that is changing. So we have new drug modalities that are starting to address the undruggable. So now, in addition to those traditionally druggable targets where there is still active pharmaceutical research, we now have new modalities, things like antibody drug conjugates, protein degraders, T-cell helpers, and others that are working in ways, chemical ways, that were not addressable previously. So they can do things like address a protein that doesn't have a well-defined binding pocket. or that's shared by multiple proteins. And they do this because they have a fundamentally different way of interacting with that protein. It's not just all about small molecules binding with a very specific part of the binding pocket, the active site. There are different ways that proteins can interact with the drug that enables the change in the disease state from the protein interaction. So, for example, here are two of those new drug modalities. Targeted protein degradation uses the ubiquitin degradation pathway. You'll see things like PROTAC or molecular glues involved, targeting the ultracellular domain. Similarly, antibody drug conjugates. This is where you put together aspects of a biologic and of a small molecule, and you attach those two. So the antibody will target a specific cell type through a particular cell surface receptor, and it will deliver the small molecule warhead directly into that cell type and only that cell type. So now you have a number of different ways where you can interact with proteins at a level far beyond that narrow number of targets. And what this results in is a tremendous expansion in the number of proteins that are now druggable. So instead of only those 900 where we have had pharmaceutical traction in the past, we're looking at tens of thousands of potential protein targets. And it fundamentally changes what we can go after as a pharmaceutical industry. It really changes the fundamental question. So the fundamental question is no longer, how do I find a protein in the disease area that is druggable? The new question, and which is really going to usher in a completely new era in drug discovery, is of all of the proteins involved in an indication, involved in a disease, how do you identify and validate a novel target? So what is the optimal protein to treat a disease? No longer what is the most tractable or amenable protein within that disease set. Fundamentally different question, which we think is going to fundamentally change the industry.
Thank you so much, Jonathan. So just to introduce myself, my name is Mo Jain. I've spent the last 20 years in analytical chemistry and as a physician, and really have the pleasure of leading the Sapient team here as we move forward. And as Jonathan mentioned, there's tremendous amount of additional inertia that's coming from the fact that the space for which we can drug now has greatly, greatly expanded from 900 targets to 4,000 proteins to now theoretically every single protein, every single proteoform that's present in humans. So we have to take a step back and just think about, how do we now go about identifying what is the ideal target? No longer what is the most druggable target, but now what is the most ideal target? Now, to date, much of target identification and validation has really centered around genomics. And there's practical reasons for this. There's been a next-gen sequencing revolution that's occurred over the last 20 years. As everyone's aware, the amount of data that can be generated per unit cost has dropped exponentially for sequencing. And because of this, there's been a tremendous amount of focus about leveraging sequencing, particularly at a genomics level and at a transcriptomics level through GWAS and RNA-based studies in order to identify the key targets. Now, when we think about the canonical pathway by which DNA gives rise to RNA, gives rise to proteins, as Jonathan mentioned earlier, the real key here, the target for the vast majority of therapeutics, independent of the modality, if it's a small molecule, a large molecule, a gene and cell therapy, a targeted protein degrader, an ADC, almost irrespective of the underlying modality, the target is the protein itself. Now, to date, given practical constraints, we used DNA and RNA as surrogate measures to understand the proteins that may be altered in a particular disease state, and in doing so, identify the ideal drug targets. As I mentioned, druggability occurs at the protein level, and the challenge in using DNA and RNA as surrogates is that these are limited. Under most conditions of disease or drug response, the relationship that occurs between the RNA level and the protein level begins to disintegrate. Now, this comes from an exceptional review from Rudy Aebersold, thinking about the relationship between RNA expression and protein expression. And the crux of this is if we look under a steady state condition, a healthy condition, with a normal quiescent cell from one individual relative to that same quiescent cell from another individual, and we look at the relationship between the RNA and the protein levels, we see under normal circumstances, there's fairly good correlation as shown here on the left. This suggests that under healthy states, oftentimes one may be able to use RNA level as a surrogate to understand those proteins that may be present in a particular cell type. The challenge is under a perturbed state, meaning whether or not there's been disease, whether there's dedifferentiation, where there's a response occurring, or even in a treated state whereby there's a therapeutic that's been instituted, this relationship between RNA levels and protein levels becomes much more muddled. And no longer does the measurement of RNA inform on the level of proteins. Now, you could think about there's many reasons why this may occur. Well, one of the common explanations is around signal delay, where we oftentimes see an acute change in RNA levels in response to a particular perturbation, whether it be a disease state, whether it be institution of treatment, whether it be dedifferentiation, et cetera. But the protein levels are lagging behind. Now, this is for an individual protein level, and you can imagine when you multiplex this across 20,000 proteins or 100,000 proteoforms, that this delay that occurs between the RNA change and the protein change can become even more great. And this is what causes this discrepancy now between these two. And as this dissociation gets worse and worse, you can imagine it's very difficult to actually infer protein levels from actual RNA expression. Now, what's the evidence then that measuring protein levels can inform across biological insight and can reveal new therapeutic targets? Well, over the last decade, there's been introduction of a number of affinity-based proteomic methods. These essentially use binding agents, whether it be antibodies or aptamers, to bind proteins of interest and then read out using a sequence tag. As these have applied to the first large-scale studies, whether it be in the UK Biobank or in other large data sets, we are revealing very new biological insights, new therapeutic targets that were not revealed by DNA alone or by transcriptional data alone. So there's really quite a bit of evidence now that's accumulating across tens of thousands of individuals that proteomics data can provide rich biological insight into new targets and new mechanisms that are driving disease, and in doing so, reveal therapeutic insights into particular targets that may be ideal for approaching using these new modalities. Now, as with the case with all bioanalytical approaches, there are important limitations to think through when we think about these affinity-based proteomics approaches. Now, really, as I mentioned, affinity-based approaches build upon sequencing technology. And fundamentally, the way they work is by binding of the protein, either using an aptamer or an antibody. And that binding agent has a sequence tag on it that's read out then by a sequencing system, whether it be array-based, whether it be PCR, or whether it be NGS-based. The challenge with this, of course, is that the protein level then is inferred indirectly by measurement of a sequencing tag that's on the affinity binding agent. What happens here is the fidelity and the specificity of the measurement has to do with how well that binding agent actually binds that protein. And for anyone who's ever run a Western blot, we know even for the best antibodies, there's not perfect binding that occurs. And oftentimes a particular binding agent can bind multiple proteins. We know once again that the way these binding agents occur is through a single interaction or point interaction that occurs with the protein on its three-dimensional structure. Because of these antibody or aptamer-based affinity approaches do not have the ability generally to detect post-translational modifications, splice variants, or other isoform variants, which may in many ways alter the actual protein function. Because we're introducing these binding agents, you can only measure those proteins that are targeted for which there's an actual binding agent. And as you can imagine, the matrix becomes extremely important. And so most of these binding agents have been optimized for the study of human blood, which means it's difficult to study cellular systems, preclinical systems, or even within humans, the actual tumor itself or alternative biofluids. Because many of these important limitations, including around the specificity and matrix interactions, it's very difficult to operationalize these affinity-based protein measures in regulated environments, whether it be GLP or CLIA-based approaches, which means that they've been very useful for initial R&D discovery. But once translation occurs, this requires returning to a whole different modality for measurement. This is where traditional mass spectrometry now has been really quite important. As you're well aware, mass spectrometry has always offered an alternative approach for measuring of proteins. Because these work through direct peptide sequencing, whereby a protein is broken up into its peptide fragments, and those individual peptide fragments are then sequenced one by one, and the protein reconstructed, this direct measurement of the protein provides for very high specificity around the idea of a particular molecule. And so there's quite a bit of confidence when we're identifying a protein using these approaches. As you can imagine, mass spectrometry is quite complementary to affinity-based approaches and solves for many of the limitations that we've seen in affinity-based proteomics, including binding specificity through direct measurements. It allows for capture of post-translational modifications and isoform variants because of the direct peptide sequencing. Because mass spectrometry operates in a discovery mode, we can measure many, many different types of proteins and peptides, not only those for which we have binding agents. Again, mass spectrometry is applicable to many different sample matrices, whether it be solid tissue samples, such as tumors and tissues, whether it be liquid samples, such as blood, urine, CSF, tears, eye fluids, or even media, whether it be a preclinical system, a clinical system, or cellular system. And mass spectrometry, as we all know, has been operationalized for many years with the GLP and CLIA environments, so it allows for direct translation of discoveries from the R&D laboratory into actual clinical trials and clinical practices. Now, importantly, traditional mass spectrometry is not without its own limitations. For anyone who's worked in this space for the last several decades, you know that mass spectrometry is plagued by traditionally very low throughput. Mass spectrometers have traditionally been quite slow. They have limited sensitivity, which limits the depth of coverage, meaning the total number of proteins that can be captured in any single biological samples. With certain biospecimens, including plasma, the large concentration gradient between the most abundant and the least abundant proteins introduces very unique bioanalytical challenges that have traditionally, once again, limited the utility of mass spectrometry. And of course, on the back end, given the complexity of mass spectral data, they have traditionally been bottlenecks in actual data analytics and how we reconstruct proteins from actual peptide sequences. Importantly, over the last year or so, there's been massive transformation in mass spectrometry. Step gradient changes that have occurred at a number of steps that we'll review today that have ushered in the next generation of mass spectrometry, and this has completely altered the potential landscape for discovery proteomics. Critical innovations have occurred at almost every single step, from sample handling to mass spec hardware to data software, that in totality have transformed the utility of mass spectrometry. Just to review these on the left, when we think about automation, there's been the introduction of a number of automation systems now that allow us to relieve manual workflows and process samples at scale. Instrumentation, particularly around mass spectrometry, has been completely transformed, and this has greatly accelerated over an order of magnitude the rate of data acquisition. There's been very important advances that have occurred on sample processing, particularly through the introduction of nanoparticles that allow for depletion of these high abundant proteins and enrichment of low abundant proteins, and enable us to measure many, many thousands of proteins more than traditionally have been done. There's quite a bit of innovation that's occurred on the back-end software and the processing of mass spectral data, using imaging-based and machine learning-based approaches to reconstruct protein levels from peptide sequences, including those that use distributed computing and the cloud to do this at scale. And finally, as many of us heard about, there's been massive advances in AI-based data sciences, which allow us to address very critical bottlenecks in data interpretation and deriving knowledge and insight from underlying large data sets. So the results of these critical innovations have been absolutely apparent. The coverage has greatly expanded, whereby traditionally we've been limited to hundreds or very few thousands of proteins measured in a biological specimen. This has changed massively now, where we can routinely measure tens of thousands of proteins and post-translational modifications in a biosample in a true non-targeted approach. We've been able to greatly scale the throughput, whereby traditionally only a couple dozen samples could be run per day. Now this is scaled to potentially running hundreds of samples per day, per instrument, and per operator. And the robustness of mass spectrometry data, because of all these innovations, has improved drastically, which allows for rapid translatability, once again, not only handling preclinical and cellular samples, but also clinical samples across a number of sample matrices, from plasma to urine to CSF to tears to tissues to tumors to cells, et cetera, et cetera. And so in totality now, mass spectrometry has greatly emerged as a new modality that allows us to finally be able to identify targets at scale. Here at Sapient, we've spent years developing these technologies, and shown here is a video of our laboratory based in San Diego. This is a small portion of our laboratory in which we house a massive amount of mass spectrometry infrastructure. Well, we leverage a number of different types of mass spectrometers, including the Bruker, timsTOF, HT, and Pro2 mass spectrometers, which are really state-of-the-art, high-resolution QTOF ion mobility systems. These systems allow us to take any biological specimen, whether it be a liquid specimen such as blood, urine, CSF, or tears, or a solid sample, whether it be tissues, tumors, or cells in a dish, preclinical or clinical, and in that singular sample, assay thousands of metabolites, lipids, and proteins simultaneously. As mentioned earlier, we can do this under R&D designation, as well as rapidly and seamlessly translate these discoveries into our CLIA/CAP accredited laboratory, where we develop precision targeted panels, which can be readily applied as part of clinical studies or as part of clinical implementation. Just to talk through the approach here in schematics, we are leveraging a number of technologies, not only on the sample processing, but also on the mass spectrometry and the data processing side, which in totality allow us to measure more, faster, and cheaper than we've ever been able to do. When we think about liquid samples such as blood and CSF, we're leveraging nanoparticle extraction systems that have been optimized here at Sapient that allow us to take a biological specimen, deplete the super high abundant proteins that may cause artifact and interaction, and enrich for the lower abundant proteins, as well as capture both exosomal and EV proteins. And this has completely transformed the total number of proteins we can assay in a singular experiment. From a mass spectrometry side, once again, we're leveraging state-of-the-art mass spectrometry equipment, including the timsTOF HT systems, which allow us to capture not only MS2 data, but also ion mobility for label-free DIA discovery proteomics. We leverage these within the context of EvoSep chromatography to maximize stability across large sample numbers. And then on the back end, we're using state-of-the-art software that enables direct peptide sequencing and allows for protein identification with extremely high confidence, as well as to think through protein isoforms, including PTMs, splice variants, and other isoformic polymorphic variants. Within a singular plasma sample, we're catching such PTMs as phosphorylation, acetylation, methylation, and ubiquitination, all of which can be critical for altering the underlying protein function. As mentioned, these technologies have been optimized now to enable study of cellular systems, of preclinical systems, or clinical samples for both liquid as well as tissue at scale. And because we're capturing not only the free-floating proteins that may be in the blood or may be in the CSF or may be in the urine, but also the exosomal and EV-based proteins, we're capturing critically biologically important proteins, including many of the canonical drug targets that Jonathan mentioned earlier. Now, when we think about proteomics and its application, there's no one-size-fits-all. And at Sapient, we've developed multiple workflows, each of which is optimized for a different analytical characteristic. Just to walk you through this, from the left, we have what's called our deep discovery proteomics. This is optimized to maximally capture all the proteins that can possibly be measured using these instrumentation in a singular sample. And so for a typical cell or tissue sample, we're assaying over 12,000 proteins with full PTM coverage here. This is a slightly slower method, but still it provides capacity to handle thousands of samples in a given experiment. We have our core discovery method next, which is really optimized for plasma or liquid-based samples, whether it be CSF, tears, urine, or media. And in these systems, we're able to now measure north of 6,000 proteins, including in a single human plasma sample, with quite remarkable capacity to handle tens of thousands of samples in a given experiment. And as you can imagine, these approaches are really optimized to reveal biological insight from the plasma and other liquid proteomes. We have high throughput discovery methods that are really optimized for speed and maximizing the number of samples that can be assayed in unit time. And using these high throughput measures, we're capturing north of 2,000 proteins in plasma, more than 6,000 proteins in cells, again with full PTM coverage. We can handle north of 100,000 samples using these types of methodologies. And these are really optimized for the study of large population scale-based studies, as well as for cellular-based screening, as we'll talk about in just a moment. And finally, we have what's called our precision-guided approaches. This is after we've made a critical discovery, or if there's a key marker already a priori that is desired for measure, we can develop very precise and targeted methods that allow for capture of isoformic information or PTM information around singular proteins of interest, again, across a multiple of matrices, including running these in our high-complexity CLIA laboratory. Jonathan?
So Mo has taken us through a true transformation in how we characterize and measure proteins at scale. This doesn't take us to the point of transforming pharmaceutical discovery, however. There's still more needed. Just being able to measure and characterize proteins doesn't close the gap to being able to drug new targets in the treatment of disease. And what is that gap? That gap is the understanding of the disease in the context of the proteins being identified. So what we've done is develop a human biology database, which includes real-world data from over 100,000 patients. This data includes longitudinal clinical outcomes, as well as other things that would be impactful about understanding the disease state, things like lifestyle factors, or especially patient response to therapy. Therefore, you can design cohorts based on healthy versus disease, as well as different treatment arms, and segregate those by different demographic factors like age or sex or prior disease and relapse, key things that you would normally build into real-world data cohorts to understand the disease. And then you can do surveys of proteins and of other metabolites through metabolomics or through lipidomics to really understand what drug targets now in the broadly expanded druggable space you should pursue. And so if we get very tactical in terms of the pharmaceutical value chain and how this is used, here we've laid out the pharmaceutical value chain from early in drug discovery all the way through approval, going through translational. And there are a number of different use cases that are enabled by this new proteomics technology, as well as metabolomics, lipidomics, in concert with genomics, and then putting that together with real-world data assets. We don't have time to get through all of them today, but there are a number of good ones going from abnormal protein expression and high throughput drug screening in the preclinical setting, all the way through dose selection, biomarker discovery, target engagement and patient selection, and all the way up to companion diagnostics. So lots of strategies to accelerate clinical programs. Just in the interest of time, we're going to dive into two of the discovery use cases around target identification and validation and drug discovery.
So just to walk you through this, here is a real-world example by which we may be able to use these next-generational proteomic systems for target identification. Shown here on the left is a volcano plot, defining these 12 to 13,000 proteins in a disease state as well as control. In this case, these are a group of tumors, solid-state tumors, as well as their normal tissue counterparts. Here we can see from the volcano plot, in blue, there's thousands upon thousands of proteins that are altered in the setting of disease, with very high confidence p-values as shown on the y-axis. When we look to the right of the zero, these are proteins that are increased in the disease state. And interestingly enough, when we look to the left of the zero, these are a number of proteins that are decreased in the disease state. Now, if we take these thousands of proteins and map these on to known cancer therapeutic targets, what we find is here is that there's a subset of these for which there are already known targets for which drugs have been developed and are in the clinic, or emerging therapeutics are under development from these orange dots. And what you can see is that while the orange dots are heavily enriched at the extremes, there's still a number of additional protein targets for which drug development is not occurring. And it's only by looking using these comprehensive tools that we can find these additional protein targets. So how do we go through the process of validating them? And this is where additional data that comes from our data assets can be absolutely essential. And we call this process enhanced target validation. First, we can look not only in that normal tissue that corresponds to the tumor, but we also look for the expression of these proteins now across more than 20 other normal human tissues. And in doing so, we can begin to understand what may be the potential adverse effects that may come from targeting this tissue. We provide tremendous amount of evidence regarding the underlying contextualization of this particular target, thinking through the downstream biology and pathways that are impacted by this particular protein target using our data assets. And then finally, using AI-based approaches, we can begin to understand the biology and the substructure localization of these particular targets. And in doing so, we can understand the ideal modality that may be required to drug these specific targets. Now, just to take a step back and think through potential pathways that may be altered, at Sapient, we've organized the human proteome into a series of signaling cascades and pathways that once again provide biological insight. Leveraging the data that's present within our human biology database that Jonathan mentioned, we can begin to understand the alteration of particular signaling cascades and pathways that may occur from specific targets. And so shown here is an example of 40 or so signaling cascades that are routinely monitored by our proteomics approaches. So in using this information, we can not only identify a particular target, but the particular biological process and even the specific signaling cascade that may be altered by that target, which provides a tremendous amount of contextualization, not only for understanding disease mechanisms, but also potentially identifying PD markers and fundamentally understanding mechanisms by which a drug may impact disease processes. Similarly, we can use our AI-based approaches to understand the substructure and localization of particular targets, including to understand those targets which are present at the surface, what we call our cellular surface proteome. And this has very important implications for understanding particular therapeutics, particularly within the case of oncology. And so for any of these targets that we identify, we go through this process where we use our AI-based characterization to be able to define where in the cell the particular target may be localized. You can imagine for defining those that are present at the cell surface, this provides tremendous amount of opportunity for using emerging therapeutics such as ADCs, radioligand-based therapies, and T-cell engagers to engage these target antigens. As part of this process, we identify these target-specific antigens that are located at the cell surface protein, as well as define their abundance levels and quantify their actual copy number, not only in the actual tumor samples themselves, but across a host of normal cells. And this again provides information regarding the specificity of this particular target. We can also use these methodologies as part of the next step. Once we've identified the key target, to engage and accelerate and advance the underlying screening process. And for here, we can use a multitude of different analytical approaches that are in place at Sapient. Here is shown you as just one such example in which we're leveraging what's called our protein degrader proteomics approaches, in which we're using multiple different methods, including first our high throughput screening methods to screen a panel of molecular glues or PROTACs or other degrader biologies to identify those specific targets that may be altered by specific degrader systems. As we prioritize candidates, we can use our deep proteomics degrader methods to understand time points of degradation, as well as selectivity profiles, understand downstream pathways that may be altered, as well as potential adverse events and off-target knockdown that may occur from these actual degrader systems. And then ultimately, we can use our precision-guided approaches to develop singular assays for PD markers and target engagement markers that can then enroll in the clinical trials as part of the development of these assets.
So zooming out a little bit, we've focused a lot on, in this talk, on the opportunity to apply proteomics for new, previously undruggable targets, novel targets where we think that there is a very clear path to validation and pharmaceutical development. But proteomics at this scale and backed by this kind of data has applicability beyond just finding the right disease targets. An important role there, but backing up, there's not just the new target. You have to, in precision medicine, find the right patient and adjust the right dose and the right therapeutic aspects of the drug. So there's an enormous role that proteomics and data plays in the right patient aspects. So patient response and non-responsive, being predictive around that, stratifying patients so that only those patients that will have an efficacious response and not a safety issue or an adverse event are on therapy and are even recruited into the clinical trial. We can get very detailed in terms of safety profiling or the development of companion diagnostics, or ensuring that in the INEX criteria, the inclusion-exclusion criteria built into a protocol, we are enriching those trials for the patients that will benefit most from therapy, therefore increasing the probability of success on a per trial basis. And then if we look at the right therapy aspects of proteomics, a lot to be done in terms of dosing, optimal dosing, really hitting that therapeutic window and staying within the safety window for dosing. The timing of the treatments, how we engage the target, and ultimately how we build a tox profile that will translate from rodents and non-human species into the clinic for an entry into human aspect of a program development. So proteomics and data goes far beyond what we talked about today in terms of expanding the druggable space. It really impacts all of precision medicine.
We're happy to pause there and would invite the audience to please share any questions that you may have in the chat. We would like to thank everyone for their time and attention today and for the opportunity to speak with you today, and would be really excited to time allotting take any questions.
Perfect. Thank you both for such insightful presentations. So first question, you talk about going beyond the genome. Do you see genomics and proteomics data as completely independent, or is there value in integrating both data sets?
Yeah, it's a great question. I can take that one. So first of all, we're talking to you today from Southern California. And so I'm going to use a Southern California analogy to kind of explain how genomics and proteomics work together rather than be completely independent. If we think of genomics as the structure of the roads, the overall highways and secondary roads, and give you a map, proteomics is all about the specifics of getting where you want to be. So the traffic, which lane you should be in, where you should park, what time you should arrive, when you should leave. All of that is happening at the protein level in both healthy and diseased tissue or blood. Genomics is great to understand the overall structure, and much of the mapping that we do in proteomics is mapped back to an actual gene, and we use gene atlas extensively as what is the protein function. That only takes you so far. As I was talking about, hundreds of thousands of isoforms relative to a smaller number of genes, 20,000 genes. So there's just so much more context associated with things like traffic and where to park that are of practical consideration in drug development. And just knowing where the highway goes is not everything you need. Lipidomics, metabolomics, similarly give you that kind of traffic, where to park, when to leave, when to arrive type information, and are really impactful in drug development programs.
Question 2. Where do you view affinity-based methods in proteomics compared to mass spectrometry?
Thank you for the question. Happy to jump in. So we very much view these as complementary approaches, right? With any bioanalytical approach, it's important to understand the strengths and limitations. I would say traditionally, meaning several years ago, affinity-based proteomics methods were head and shoulders above mass spectrometry for the very reasons that we discussed in this webinar today, whereby there was issues with throughput and depth of data generation, et cetera, that would occur with mass spectrometry. Given the innovations that have occurred that we discussed today, proteomics, as from a mass spectrometry perspective now, has completely been transformed. I still view this as there being a complementary approach in that there are certain proteins in the blood or in a tissue that are very difficult to assay by mass spectrometry. And under those circumstances, those are better measured by an affinity-based approach, whether it be a mesoscale or ELISA method, whether it be Olink or SomaLogic or NuLISA-seq. There's many different methodologies that allow for aptamer binding and measurement, and those can be quite advantageous to complement mass spectrometry data. So in particular, when we think about very low abundant molecules that are fleeting, such as cytokines and chemokines, having a binding agent or an affinity-based approach that has an amplification of the underlying signal can be quite advantageous, and those are very difficult to measure by mass spectrometry. As a whole, though, for discovery approaches, particularly for target identification, particularly for when we want information regarding PTMs or post-translational modifications or isoformic variants, et cetera, this is where mass spectrometry has become the leader in this approach, especially now given all the innovations that allow us to generate mass spectrometry data at scale, at great depth.
Wonderful, thank you. So question number 3, can you elaborate on how proteomics can be applied to accelerate personalization of therapy? How do you go from R&D discovery to patient impact?
Yeah, I can take that one. I mean, what we're really talking about here is biomarker discovery, and that is how you differentiate patient response and how you differentiate disease state, largely through biomarkers, is predictive of disease progression or response to therapy. And what we are talking about today, we focused a lot on drug discovery and especially novel targets, but proteomics has a huge role to play in the characterization of biomarkers. And if you look at healthy patients, there are literally hundreds of thousands of changes just between healthy, normal individuals. And if you go beyond proteomics to other analytes that you would measure in metabolomics or lipidomics, you're up into over millions of changes, potential measurements you could make that would distinguish these patients. Then you add in disease and disease progression, the variability of disease, heterogeneity of response to therapy. You get into a real combinatorial problem. So one of the key benefits right now of proteomics is two of them. Let me just highlight two that Mo had mentioned. One is the high throughput nature of it. The automation enables us to run many more samples and isolate and get higher N. So the statistics enable us to isolate specific biomarkers. And the second one is the degree of machine learning and other data that we are able to bring in to contextualize these biomarkers. So now you've gone down from millions of changes between patients to just a handful that are disease associated, that are predictive of patient response and that have a biological mechanism behind them that can really drive a therapeutic program.
Perfect. Thank you, Jonathan. Question number 4. So can you give some examples about how Sapient's data assets can be used to inform on target identification?
Sure, let me take that one also. The key thing with real world data assets is that you can't just have disease in there. You need to have something to compare it to. So it's really important that you have healthy normals, and that the design of the healthy cohort is absolutely critical because it needs to match the diseased cohorts in all respects: height, weight, age, demographics, even things like income level, other diseases, lifestyle factors. These things have to match up as closely as possible so that you can really identify just those things that are changing with patients that are directly disease associated. You need a lot of patients, therefore, and you need deep data on those patients. Then the next thing is there are a number of different TAs. We talked a little bit about oncology today, but breathtaking advances in neurology, in cardiometabolic, in rare disease. So it's really important that your data set not only have liquid and solid tumors, longitudinal data, but also other TAs, and then a good ability to build these cohorts to compare disease set or cohort with a normal or healthy cohort.
Thank you. So we have question number 5. Can you talk about AI and how you're integrating that into proteomics studies, especially with discovery proteomics, where so much data is being generated?
Yeah, happy to take this one, and thank you for the question. So as you can imagine, there's been a tremendous transformation that's occurred around the world in algorithmic analysis, what we call AI, that allows us to take very complex information and identify patterns from which we can make sense of that information, either for diagnosing individuals, for understanding outcomes, for stratification, et cetera. Now, there's a number of areas for which machine learning or AI is being applied within the proteomic space. Very simply put, we talked about some of the innovations that are occurring at the software level, whereby we can take these peptide sequences and from that use machine learning to reconstruct the protein levels as well as the particular isoform variants. And that's a very tactical, if you will, application for machine learning and AI. At the same time now, there's quite a bit of a shift that's occurring in the AI space, whereby it's no longer the limitation I should say, is no longer the algorithmic work or the computational infrastructure, but rather it's the data that's being inputted into the underlying AI models becomes the limitation. And you can imagine that taking the best predictive AI algorithm and putting in junk data doesn't yield any key results. And it's once we can marry the best algorithms with the best data that real insight emerges. We finally, as you heard about today in the webinar, can generate proteomics data at scale, at great depth across thousands of biological specimens, measuring tens to hundreds of thousands of potential variant isoforms of these proteins. When we combine this data with other assays that are operational and used here at Sapient, including metabolomics and lipidomics, transcriptomic profiling, or even genomics data, this is where AI can really begin to work its magic and do multidimensional integration in a way that allows us to identify years before the onset of disease who's at risk for particular diseases, whether it be neurodegenerative diseases, cancer, cardiometabolic disease, diabetes, etc. We can also use this information now to understand complex mechanisms of action that may be occurring from drugs, or even panels of targets that may be employed, or predictors of responders, non-responders. So as you can imagine, as the data that we're able to generate continues to scale every day, being able to handle this very complex data using these AI-based algorithms is absolutely essential and is playing a key role in going from data to actual knowledge to translational insight.
Thank you very much. So another question from the chat. Can you talk about where you think proteomics is going in the near future in terms of discovery biology? How is the market evolving?
Yeah, it's a great question. And maybe I could pick up on a theme that Mo was just speaking about related to AI, because a key part of the market evolution is AI is very real, and especially machine learning, which has been with us for some time, is very real. At the same time, recently, really over the last 18 months to 2 years, there has been a lot of excitement around generative AI and other types of AI that have gotten the attention of key industrial decision makers, really across industries, not just in pharma. Pharma decision makers correctly have always been very risk averse for two reasons. One, they are responsible for developing new medicines for patients with very serious unmet medical needs, right? So you have to take a very deep, disciplined approach, and you have to understand what your data is doing in order to impact the patient. The second reason is that they are often responsible for large budgets, relatively large R&D budgets, and so they want to put those resources where it makes the most sense. So I think a big aspect of the excitement in AI has enabled these pharmaceutical decision makers to really understand the value of data, which everybody did, but now the value of large data sets that are clean and that are comparable across experiments. And so much of the detail that we went into today about how we produce proteomics data in an unbiased way that can be generated in very large quantity across a large number of proteins at a very reasonable cost, that is playing directly into market evolution of proteomics in discovery biology. It's a critical point that I think it is now appropriate to invest in large protein data sets because of this technology and because of the underlying linkages that we can make to disease.
Perfect. Thank you very much. Unfortunately, that is all we have time for. If you had any questions that we didn't get to, don't worry. We will be sending these to the presenters after the webinar. So thank you very much once again to both presenters for joining us today. And as mentioned, the on-demand recording will be made available over the coming days. Thank you very much.
Thank you to Oxford Global, and thank you to the audience. I would invite anyone who's interested to visit our website at www.sapient, sapient.bio, or to reach out to us at any point via our email, our call contact, or to schedule a meeting with us to talk further if there's of interest.
Yeah, thanks very much. I really appreciated the discussion.